Complement system, an important component of the innate immune system, and also contributes to adaptive immunity, is what we're going to be looking at today. So what is the complement system? The complement system is simply a network of glycoproteins found in the serum produced in the liver that work together to enhance the activities of antibodies or phagocytic cells. The name complement, it means to enhance. They enhance the activities of antibodies or phagocytic cells to eliminate pathogens. What happens in the complement system? First and foremost, we know there are a group of about 30 glycoproteins. That is one. About 30 glycoproteins. And the major complement proteins are C1 to C9. And the complement system happens through three major pathways. The first is the classical pathway. The second is the alternative pathway. And the third is the mannose binding lecithin pathway. Now, this classical pathway of the complement system is usually activated by antibody binding. The alternative pathway is usually activated by microbial surfaces like microbial cell wall or patterns on the microbes that can be recognized by your immune system. Now, the mannose binding lecithin pathway is usually activated by microbial polysaccharides. All of them has different activation pathways, but they all converge at one point, the activation of the C3. For the purpose of this video, we are only going to be looking at the classical pathway. The classical pathway. We have said it that the classical pathway is activated by the binding of antibody to the antigen of a pathogen. The antibodies involved in this reaction can either be IgG or IgM. Don't forget that. IgG or IgM, the binding of IgG or IgM to the antigen of a pathogen activates the classical pathway of the complement system. When this bind, right, the C1 becomes activated. Now, this C1 has three parts: C1q, C1r, and C1s. Once the C1 becomes activated, this C1q goes and binds to the Fc region of the antibody. The binding of the C1q activates C1r, which in turn activates C1s. Remember that the activation of one triggers the activation of the other. That is how it happens in the complement system. It's a cascade of reactions. Now, this C1r and C1s are both proteases. So when C1s is activated, it goes and cleaves C4 into C4a and C4b. In the complement system, the A is usually the smaller fragment, while the B is the larger fragment. And it's the B that stays behind to form the reaction. So this C4A is thrown back into the serum, while this C4B goes and binds on the microbial surface. The activation of this C4B leads to the cleaving of the C2 into C2A and C2B. And C2B. Remember that we have said that the larger fragment is usually the B and the smaller fragment is the A. But for the C2, the larger fragment is the A and the smaller fragment is the B. So the C2B goes here, back into the serum, while this C2A attaches to the C4B. And these two now forms what we call the C3 convertase. It becomes the C4B and the C2A. It's called the C3 convertase. Why? Because it's what we cleave C3 into C3A and C3B. The reaction continues. This C3A goes ahead to become, it's an inflammatory molecule. It enhances inflammation. It's also an anaphylatoxin. and it can also serve as an opsonin to enhance phagocytosis, the C3A. Then the C3B goes ahead and binds to the C4B and the C2A. So it becomes C4B, C2A, and C3B. This is known as the C5 convertase. Are you seeing that the reaction of one triggers the activation of the other? So two major things that you should be aware of. The C3 convertase is the C4B and the C2A. The C4B and the C2A are the C3 convertase. While the C4B, C2A, C3B is the C5 convertase. Their work is to cleave the C5 into C5A and C5B. C5 moves down into the surrounding tissues. Also, is doing the same work as C3B. It's also an inflammatory molecule and anaphylatoxin, enhances inflammation. Why this C5B now goes ahead to bind the C4B, the C2A, the C3B to become the C4B, the C2A, the C3B, and the C5B. This initiates the formation of the membrane attack complex. Remember that the purpose of all of this reaction that is being triggered is to enhance the destruction of the pathogen. So when this C5B binds and the membrane attack complex has been initiated, the C6 is also cleaved into C6A, C6B. Goes the same for C7, C8 up to C9. Now they all become C4B. I'm coming. Sorry, my marker. They all become C4B, C2A, C3B, C5B, C6B, C7B, C8B, C9B. All of them is known as the membrane attack complex. What is the function of this membrane attack complex? They punch holes in the membrane of the pathogen, which ultimately leads to cell lysis and cell death. So you see why we said that the complement system enhances the activity of the antibody and phagocytic cells. Major points you should know. The antibodies that are involved are IgG or IgM. The classical pathway is activated by the binding of antibody to the antigen. The C3 convertase is the C4B, C2A. The C5 convertase is the C4B, C2A, and C3B. And regardless of the pathway in the complement system, the difference in the pathway is what triggers their activation. Like we have said, the classical pathway is triggered by the binding of an antibody. The alternative pathway is antibody independent, is triggered by the presence of microbial surface or components of the microbe, either the cell wall or receptors. While the mannose lecithin pathway is triggered by microbial polysaccharide. So regardless of what activates it, once it gets to the activation of this C3, the reaction becomes the same for all of the pathways, because the ultimate goal is one, to form the membrane attack complex, which is to puncture holes in the membrane of the pathogen. So you understand now that the complement system, a part of the innate system, and also contributes to adaptive immunity. In the next video, we are going to be looking at the alternative pathway and the mannose lecithin pathway. But don't forget, once you know the larger fragments, the smaller fragments, you know what forms the reaction. See you in the next video.